Image 1. Parthenocarpy, polyembryony, apomixis. Parthenocarpy, 1.14. This section explains fruit development without fertilization, leading to seedless fruits, e.g. pineapple, banana, papaya. It mentions the role of hormones like IA, indole 3-acetic acid. Polyembryony, 1.15. This describes the development of more than one embryo inside a seed. It can be true polyembryony, e.g. Nucella polyembryony in citrus, or false polyembryony. True polyembryony often involves nucella cells developing into embryos. Think about it / do you know? These sections pose questions about polyembryony in citrus and clarify that parthenogenesis is the development of an embryo directly from an egg, which is a kind of apomixis. It also mentions different ways polyembryony can be induced or occur naturally, e.g. genetic, foreign pollen, fertilization of nucella cells. Activity suggests preparing a chart for natural vegetative propagation, indicating the vegetative part / S and examples. Image 2. Embryo development, dicot and monocot. 1.11 Development of embryo explains that the process of development of zygote into an embryo is called embryogenesis. The embryo develops within the embryo sac. Fig 1.15. Development of dicoembryo in capsella. This diagram illustrates the stages of dicoembryo development, showing the zygote, proembryo, globular stage, heart-shaped stage, and mature embryo, with suspensor, radicle, plumule, cotyledons. It describes how the proembryo divides to form the terminal cell, which forms the embryo, and the basal cell, which forms the suspensor. Fig 1.16. Development of monocot embryo. This diagram shows the development of a monocot embryo, highlighting structures like the coleoptile and scutellum. The text describes the suspensor and haustorium in monocots. Image 3. Apomixis and seed fruit formation. 1.13 Apomixis. This is a general term for asexual reproduction without fertilization, producing offspring genetically identical to the parent. It covers different types of apomixis. Recurrent apomixis, where the embryo sac forms from an archosporial cell or some other part of the nucellus, leading to a diploid embryo sac. Diplospory, nucella cells form embryo sac, and apospory, nucella cells form embryo sac, are mentioned. Non-recurrent apomixis involves meiosis and a haploid embryo sac, with the embryo forming from the egg without fertilization. Adventative embryony. Embryos develop directly from somatic nucellus or integuments without fertilization, e.g. mango, orange, lemon, citrus. Can you recall / significance of seed and fruit formation? These sections likely ask questions about the parts of a fruit and the importance of fruits and seeds for plant propagation and dispersal. Image 4. Monocot seed and dicot seed. Fig 1.18. Maize seed, monocot. This diagram shows the structure of a maize corn seed, highlighting the pericarp, endosperm, starchy part, scutellum, cotyledon, plumule, and radicle. The text explains that the maize seed is a fruit with a fused pericarp and seed coat. Fig 1.17. Bean seed, dicot. This diagram illustrates a bean seed, showing the two cotyledons, plumule, and radicle. 1.12. Seed and fruit development. This section generally describes how ovules develop into seeds and ovaries into fruits after fertilization. It also mentions that endosperm provides nourishment to the developing embryo, and that non-endospermic or albuminous seeds, like bean, store food in cotyledons, while endospermic seeds, like maize, store food in the endosperm.